Chapter 9 Advanced Linkage Analysis Part 2 Specialized Mapping Technique Linkage Analysis in Humans. Mapping genes and human pedigrees. Methods of rcDNA are used to map human chromosomes and locate genes, and we can clone them to determine structure and function. Human pedigrees and DNA mapping are used to identify dominant and recessive disease genes, but for human genetic mapping, we use polymorphic DNA sequences. What is polymorphic DNA sequences? The presence in a population of two or more relatively common forms of gene in a chromosome is called polymorphism. Changes of DNA fragment length produced by presence and absence by cleavage and DNA molecules are known as Rflp, decision fragment length polymorphism. Prevalent type of polymorphism is single base pair difference. We call it SNP, simple nucleotide polymorphism. A genetic polymorphism resulting from a randomly repeated short DNA sequence is called a SSR, simple sequence repeat. So we use it for parentage testing and density determination. So these are the three types of polymorphism you can see. Rflp, you can see the glowy thing on the right, the glowy thing on the left. SNP, you can see that all of these sequences are look for differences between them. SSR will look like if any repeats happen in between the sequences. First of all is the Rflp. Rflp is from the word precision fragment length polymorphism. Precision endonuclease enzymes are used to map genes because they produce a unique set of fragments for a gene. There are more than 200 precision endonucleases in use, and each recognizes a specific sequence of DNA bases. A chloride cuts double-stranded DNA at the sequence 5 GAATC 3 wherever it occurs. So here you can see it is a long DNA, but if we treat them with E. coli, any place where there is a GAATTC, CTTAG, E. coli will cut there because it is the E. coli cleavage site. So when it cut, then the DNA will become fragments. Differences in DNA sequence generate different recognition sequences and DNA sequence sites for specific reaction enzymes. Two different genes will produce different fragment patterns when cut with the same reaction enzyme due to differences in the DNA sequence. So in the picture below, you can see this DNA treated with E. coli. Okay, this DNA has three reaction enzyme sites that E. coli can cut. So now you have two fragments. But in this DNA, the DNA doesn't even have a reaction enzyme site because in the middle there, GACTC. So it is being mutated. Because of this being mutated, E. coli cannot cut. So there is no cleavage. We have one larger fragment. So Rflp, if you want to consider using it, it is slow process, uses red isotopes. But nowadays you can even find the one that is not red isotopes because it can create radioactivity. It might poison you. You also need high quality DNA and polymorphic probes, so it might be expensive, and codon markers often specific to species. Where do we apply Rflp? We can use it to identify species or populations, estimate genetic distance, fingerprinting, forensic, or who is the father, disease status, genetic mapping, and also the relation between taxa or types of animals. You know, taxa, you have a species, genus, family, order. We want to look at how similar or how different these animals are. SNPs, pronounced SNP. It is genetic variation when one single nucleotide is altered and carried through heredity. So you have ATTG, then it becomes ATAG. So in the human genome, it is very abundant. Human genome has 3 million SNPs, but it occurs in more than 1% of population, and most SNPs are biallelic. So you have two variations. So as you can see here, this is 3 people. Okay, you have GCAA. This blue guy is GCAG. Yellow guy is GCAT. So where is the SNP? The SNP is in the fourth letter, AGT. That's the area where it is changed. So the total of number of SNPs is 1 to the power of 10 to the power 7. So difference between two individuals, which is 3 to the power 10 of 6, meaning that it is 0.1% of the entire genome. Single nucleotide polymorphism SNPs are the most common type of genome variation. So in your genome, it happens a lot, but in this example, we show you just one: AAT, CG, TAC, and then below we have AAT, CGT, GTAC. What is the difference? The difference is a single nucleotide in the number 6. So the top has C, the bottom has T. So it is a change in a single pair of vascular position. As everyone here has a copy of this region of sequence for each parent, a person can have one of 3 patterns of this SNP, CC, CT, or TD. To be called an SNP, the base change has to have a frequency of more than 1%. Challenges: you need to test so many of them, and for a single SNP test, you need to have millions. That about 10 tests is needed for SNP pair-based test. And because of this, we need to maintain statistical power and reduce false positive regression, from error. Less alleles, so each marker is less informative. Misinterpretation, much more difficult. And multiplexers do not actually work yet. So it currently uses more DNA sample than short tandem repeats use. Advantages: pizza products can be very small. Markers can work with extremely degraded DNA samples. It is more common in the genome and may possibly multiplex hundreds and thousands in one chip in the future. Sample processing can be automated and there will be no starter products. SSRs, simple sequence repeats. SSRs are DNA sequences with repeated lengths for a few base pairs. When DNA molecule cleave with shared nucleus, the cleave at sites flanking the tandem repeat. Size of DNA fragment produced is determined by the number of repeats present in the molecule. As you can see on the picture on the right, the first you have just one CA, the second is CACA, you have 2 repeats, then you have CACA, 3. The more repeats that you have, means that the DNA fragment will be even bigger in size. So there is an average of 1 SSR per 2 kilobase of human DNA, meaning that after 2000 bases, you have 1. The number of SSRs is highly variable among individuals. So you can see this is the unique flanking regions. The part where you can see these arrows, the blue arrows, indicates number of SSR, and the SSR will make the area longer or shorter. Advantages: requires very little, not necessarily high quality DNA, and it is highly polymorphic. It is evenly distributed throughout the genome, not just like the one before, every 2 kb, very very far away, and then you have one. And then you have simple interpretation of the results, easily automated, allowing multiplexing, meaning that you can do many batches at the same time, and you have good analytical resolution and high reproducibility. Application for individual genotyping, parentage, genetic diversity, population genetic study, genome mapping, evolutionary study like hybridization, mapping genes in human pedigrees. One source of the utility of SNPs and SSR in human genetic mapping is the high density across the genome. Additional source of utility of SSRs in genetic mapping is a large number of alleles that can be present in any human population. Mapping genes in human pedigrees. The pedigrees can be analyzed for the inheritance pattern of different alleles of the gene based on differences in SSR and SNPs. Precision enzyme cleavage of polymorphic alleles that are different in Rflp pattern produces different size fragments by gel electrophoresis. A rare recessive disease that affects people late in life is 90% linked to the Rflp marker in the gel low. What is the probability that 2-1 is the carrier of the disease? 100%, because he carries the bands indicating that that's the marker of the gene that carry the disease. What is the probability that the grandchildren grow 3 are carriers? Okay. So as you can see, 90% is 1-4, 90% is 3-4. Okay, why are they classified like that? Because they actually have the same DNA fragment that is observed in the grandmother. What is the probability that 2-1 is the carrier of the disease? So you have the markers there, 100%. What is the probability that the grandchildren are carriers? Right now it is reversed. Anybody that has that DNA marker, which is similar with the grandfather, have the disease. So now we move to Tetrad Analysis. In Tetrad Analysis, fungi will create this cellular structure called ascus, which has tetrads. So you know that in meiosis, one cell will create 4 daughter cells, and in tetrad, you have all daughter cells in one ascus. Okay, so each product of meiosis is a spore called ascospore, and all of the ascospores form the one meiosis cell, remain together in the ascus. In that, one of the ascus would have all of the possibilities of the meiosis. Several features of the ascus producing organisms are especially useful for genetic analysis. So the haploid, so the genotype will express directly in the phenotype. Not like us, we are diploid. If we have heterozygous, probably the phenotype will not be seen. But because they are haploid, even one allele is enough to produce that phenotype. Also, fungi produce very large numbers of progeny. You know, one fungi can create thousands of spores, just like powder, and their life cycles tend to be short. In tetrads, when 2 pairs of alleles are segregating, 3 patterns of segregation are possible. You have the PD, parental ditype, meaning that you have 2 parental genotypes. NPD, non-parental ditype, meaning that you only have recombinant combinations. Lastly, tetratype, TT, all 4 genotypes are observed. Look at the example on the right. AA, BB, and then small a, small b, small b. If there is no crossing over, you will have a parental ditype tetrad, meaning that the spores will be just like the parents, big A, big B, big A, big B, small a, small b, small a, small b, right? What if you have small a, small b, small b, they assort in a different way. So you'll have big A, small b, big A, small b, small a, small big B, small a, big B. So these are the non-parental type tetrad. What if you have crossing over between one of the genes and its centromere? So you have tetratype tetrad. The part where it is crossover will exchange. So you will have what? You have big A, small b, or small a, big B. But the other two will remain just like the parent. Big A, big B, small a, small b. Okay? Same goes with the other one. If the other one, which is B, also crossover, A still remains. But B, you can have big A, small b, or small a, big B. All of these will be expressed in the tetrad. When genes are unlinked, the parental ditype tetrads and the non-parental ditype tetrads are expected in equal frequencies, PD and NPD. Linkage is indicated when non-parental ditype tetrads appear for much lower frequency than parental ditype tetrads, PD and PD. Meio distance between 2 genes that are sufficiently close that double and high levels of crossover can be narrated equals half times the number of TT over the number of tetrads times 100. That's what you get for map distance. This is a neurospora crassa. Neurospora crassa creates ordered tetrads, meaning that each product of the meiosis will be created one after another. Ordered asci can also be classified as PD, NPD, or TT with respect to 2 pairs of alleles. So means that you have not just one but 2, AA, BB, right? So it makes it possible to assess the degree of linkage between genes. Arrangement of meiotic products is ordered, which makes it possible to determine the combination of frequency of particular gene and its centromere. In ordered tetrads, homologous centromeres of parental chromosomes separate at the first meiotic division. The centromeres of sister chromatids separate for the second meiotic division. When there is no crossover between the gene and centromere, the alleles segregate in meiosis 1. A crossover between the gene and centromere delays segregation alleles until meiosis 2. The map distance between gene and its centromere equals half, that's number of asci with second division segregation, over to the number of asci times 100. So that's the map distance. The formula is valid when the gene is close enough to centromere and there's no multiple crossovers. Gene conversion is a process by which one DNA sequence replaces a homologous sequence, such that the sequences become identical after the conversion event. Gene conversion can be either allelic, meaning that one allele of the same gene replaces another allele, or ectopic, meaning that one paralogous DNA sequence converts another. Normally, an organism that has inherited different copies of a gene from its parents is called heterozygous. This is generically represented as a genotype, big A, small a. No, you have one copy of variant A and small a. When a heterozygote creates gametes by meiosis, the alleles duplicate and end up in 2:2 ratio. 2 has big A, the other 2 has small a. We have 4 cells which are direct products of the meiosis. In gene conversion, a ratio other than the expected 2 big A, 2 small a is observed, such as 3 big A, 1 small a, and 1 big A, 3 small a. 3 big A, 1 small a means 3 times as many big A alleles as an alleles expressed in the daughter cells. So it is frequently accompanied by a combination between genetic markers on either side of conversion event, even when the flanking markers are tightly linked. It results from a normal DNA repair process in cell known as mismatch repair. Gene conversion suggests a molecular mechanism of recombination. So, the difference between allelic gene conversion and non-allelic ectopic gene conversion. So, allelic gene conversion happens during meiosis. The non-allelic occurs during meiosis, but the mechanism of repair of doubles and the breaks caused by DNA damage. In allelic gene conversion, homologous recombination between heterozygous site is the same as the mismatch in base pairing. But in non-allelic, it is usually repaired using sister chromatin of the broken duplex, not homologous chromosome, so no allelic conversion. In allelic gene conversion, it causes one allele to be converted to the other. But non-allelic doesn't have to be based on another allele. It can occur between homolog sequences at different loci from previous gene duplications. Allelic gene conversion will result in non-Mendelian segregation of genes, but non-allelic or ectopic will create evolution of gene families. So the first one, A, is a non-allelic or interlocus gene conversion, in that the conversion is a mismatch. Okay, we have blue and orange. During recombination, usually the orange will cross over with the orange. But in this case, there is a misalignment. So part of the blue is swapped with part of the orange. And then after the mismatch, now you have the genes which are imbalanced. You have blue and yellow for top. And then for bottom, you have blue, yellow, but inside of the yellow, there is part of a blue a little bit. Okay. B. B is interlocus gene conversion in cis, not trans. Meaning that you are looking at exchange between genes on the same chromosome. Unlike the one in A, where one gene is exchanged with the gene that is facing it, this one is converted by the gene within the same chromosome. But for C, C is interallelic gene conversion on homologous chromosomes. In this case, we have two alleles for the same locus, like big A, big B, right? So imagine that in between them, there's actually an exchange between them. Maybe you have big A, and then the big A exchange, now you have small a. So this happens in the same locus and within the same gene. Conversion is often due to base mismatch repair during homologous recombination. If one of 4 chromatids during meiosis pair up with another chromatid, as can occur because of sequence homology, the next strand transfer can occur follow a mismatch repair. This can alter the sequence of one of the chromosomes so that it is identical to the other. Chromatin recombination is initiated through the formation of a double strand break. So what causes a double strand break? A lot. You expose yourself to UV, you expose yourself to radiation, or maybe you drink bleach, so the bleach superoxides come to your cell and it breaks your DNA. So a lot of things can form double strand break in your DNA. So when this happens, the 5 ends of the break are then degraded, leaving the long tree overhangs of several hundred nucleotides. So your DNA must be repaired. So usually your DNA will repair based on the opposing strand. The opposing strand will be used as template to repair the missing strand. But what if one of the single 3-stranded DNA invades a homologous sequence on the homologous chromosome? Right. So when they do the crossover, the repair is not happened yet. Right. The repair hasn't been finished, but there's crossover. So the DNA repair mechanism will be like, okay, I will use this existing strand that is here to be the template for the new one. So what happens that you can see in the picture, blue is invading that empty space between the red. So the DNA repair mechanism, we use the blue as a template to bridge the red. So what happens? And now you have blue chromosome with part of the red and the red chromosome with part of the blue. So you have an intermediate. At various steps of the recombination process, a heteroduplex DNA is formed. When the mismatch occurs in heteroduplex DNA, sequence of one strand will be repaired to bind the other strand with perfect complementarity, because you know ATCG. So it causes one conversion of a sequence to another. So the repair process can follow either of the two alternative pathways. One is the DHJ, double holliday junction, causing exchange of DNA strands, and pathway 2, SDSA, synthesis dependent strand annealing. So there is information exchange but no physical exchange. Pathway 1. Gene conversion may occur associated with the physical connection of DNA replaces the two sides of the DHA. Pathway 2. Gene conversion will if the two DNA molecules are heterozygous at the site of the recombinational repair. One or two ways to resolve the resulting structure, known as holliday junction, leads to recombination, but the other does not. The breakage and rejoining is an enzymatic function carried out by the enzyme called the holliday junction resolving enzyme. Evolutionary importance. This contributed to our understanding that the adaptive function of meiotic recombination. It is also an adaptation to DNA repair. It maintains boundaries between species. Gene conversion is a network to link the DNA sequences between individual members of a species, and it homogenizes and maintains DNA sequence integrity of the gene pool of species so that it will not be tainted with other species DNA. Human genetic diseases such as congenital adrenal hypoplasia, adrenal is adrenal gland, hypoplasia is growing too big. So it is a gene conversion resulting in mutation of CYP21A2 gene. So in this picture, you can see this is a patient with a congenital adrenal hypoplasia. Congenital means that it happens in the womb. Adrenal is adrenal gland. Hypoplasia means that it grows too big. And so this female patient, genotypically female, it presents at birth with ambiguous genitalia, meaning that her genitals are neither female or male. So you have a vagina and then there's a penis going up from the middle. The severity of the abnormalities varies considerably. In this case, the clitoral hypertrophy means that the clitoris becomes too big, and partial fusion of the labioscrotal folds means that the lips of the vagina is closing. The end.